Hi there, my name is Alexandra Ransom, and I'm a recent biomedical biology graduate of Laurentian. Today I'll be presenting the work that I've done under the supervision of Dr. McLean for my fourth-year undergraduate thesis. My study investigated the effect of both doxorubicin and exercise on the formation of nitric oxide in skeletal muscle. I'll begin with giving a bit of background regarding doxorubicin and the role of oxidative stress and nitric oxide in skeletal muscle damage. I will then discuss the work that I've completed. Doxorubicin is a commonly used chemotherapeutic agent used to treat several cancers. Its action is through DNA intercalation, the inhibition of DNA topoisomerase II, and the production of reactive species. The most common side effect of DOX is its cardiotoxic effects. However, it also has detrimental effects on skeletal muscle. Even on limited doses, patients can experience debilitating muscle weakness and fatigue, and these symptoms can persist even after the cessation of chemotherapy treatment. Therefore, it's important to understand the mechanisms by which DOX causes damage to skeletal muscle. There are several proposed mechanisms by which DOX may contribute to myotoxicity. Firstly, DOX has been shown to increase the amount of reactive oxygen species emitted by mitochondria, which leads to skeletal muscle cell death. The structure of DOX contains 4 rings, one of which is an unsubstituted quinone and therefore has the ability to form what is known as a redox cycle. The redox cycling results in an excess production of reactive oxygen species, including superoxide and semiquinone radicals. When the oxidative stress exceeds the antioxidant capabilities of skeletal muscle, the disequilibrium causes damage to skeletal muscle fibers and to mitochondrial metabolism. The superoxide produced by redox cycling can then preferentially react with other biological molecules, such as nitric oxide, to form reactive nitrogen species. Nitric oxide is an important biological molecule involved in a variety of processes such as blood flow regulation. It's synthesized through 3 nitric oxide synthase isoforms, including ENOS, NNOS, and INOS. It's important to note that DOX binds directly to ENOS, shifting its production to superoxide instead of nitric oxide. It has also been suggested that DOX upregulates INOS activity, causing an excess of NO to be produced. Therefore, an increase in availability of both superoxide and nitric oxide will have implications in oxidative stress and ultimately skeletal muscle damage. Here's a schematic outlining some of the mechanisms by which DOX may contribute to myotoxicity. Interestingly, exercise may have the ability to limit skeletal muscle damage caused by DOX. Exercise increases the body's antioxidant defenses, as well as increases the production of cytoprotective proteins. The upregulation of these things can combat oxidative stress, and therefore may have the ability to decrease damage done to skeletal muscle. The purpose of this study was to determine how acute exercise following the administration of DOX influences nitric oxide concentration in skeletal muscle. We hypothesized that exercise following DOX administration would limit oxidative damage by decreasing nitric oxide levels in skeletal muscle. Therefore, we predicted to see lower levels of NO in the skeletal muscle of exercise rats. For this study, we used a total of 30 male Sprague Dawley rats, which were randomly assigned to 5 experimental groups. As seen in this diagram, the exercise groups underwent an acclimatization protocol. The acclimatization protocol took place over a 2-week period. The rats that completed the acclimatization protocol swam for 30 minutes on days 6, 9, and 12 of the 14-day period. This was done so that the rats had 48 hours between each swimming session. The swimming protocol consisted of the rats being placed in water for 30 minutes with 4% of their total body weight on a harness. All rats received an intraperitoneal injection of 4.5 mg per kilogram DOX, using a 23-gauge needle. Rats in Group A did not complete the acclimatization protocol and were sacrificed 24 hours after DOX administration. Rats in Group B underwent the acclimatization protocol, exercised for 60 minutes 24 hours following DOX administration, and were immediately euthanized after exercise. Rats in Group C underwent the acclimatization protocol, exercised for 60 minutes 24 hours after DOX administration, and were euthanized 48 hours post-DOX administration. Rats in Group D underwent the acclimatization protocol and exercised for 60 minutes 24 hours as well as 48 hours post-DOX administration. They were euthanized after their second exercise bout. Rats in Group E did not complete the acclimatization protocol and were euthanized 48 hours post-injection without any exercise. The red gastrocnemius, white gastrocnemius, plantaris, and soleus muscles were excised from the hind limbs of all rats immediately following euthanasia. The samples were frozen in liquid nitrogen and stored at -80. The samples were then freeze-dried, and the wet-to-dry weight ratio of each sample was calculated to ensure adequate freeze-drying. After freeze-drying, each muscle sample was powdered using a scalpel blade and 2 sets of blunt-end tweezers. Any visible connective tissue was teased out of the samples, and 3 to 5 milligrams of each sample was mixed with 200 microliters of 0.5 molar perchloric acid. The samples were vortexed and centrifuged, and 5 microliters of the supernatant from each sample was used for our nitric oxide analysis. We used the Model 280I nitric oxide analyzer by LG. A duplicate was ran for each sample. The concentration of nitric oxide in each sample was determined by comparing the area under the peaks to a standard curve, and then the average concentration of nitric oxide was determined for groups A through E for each of the 4 muscle types. A D'Agostino-Pearson normality test was used, and the data were found to be normal. A one-way ANOVA was used to determine significance, and a post-hoc analysis included a Tukey's multiple comparison test. These graphs represent the nitric oxide levels across the 4 muscle types. A consistent pattern of lower nitric oxide levels in groups B, C, and D, the exercise groups, are seen across the 4 muscle types. Groups B, C, and D exhibited fairly similar nitric oxide levels across the 4 muscle types. In the right plantaris muscle, Group A showed a concentration of 31.5 micromoles per milligram, and Group E showed a concentration of 25 micromoles per milligram, while Groups B, C, and D exhibited concentrations of 18, 17.5, and 17 micromoles per milligram respectively. Groups A through E were combined to make this graph, and it can be seen that nitric oxide levels are significantly lower in the gastrocnemius muscles as compared to the soleus and plantaris muscles. This result could be contributed to fiber type differences, considering the slow-twitch fibers found in the soleus and plantaris muscles contain more mitochondria and myoglobin. The conclusion of this study was that exercise groups exhibited a decrease in nitric oxide levels across all 4 muscle types. Additionally, we found that muscle types containing a higher percentage of slow-twitch muscle fibers exhibited higher levels of nitric oxide than fast-twitch muscles. This is likely due to a larger proportion of mitochondria in slow-twitch muscle fibers. These patterns suggest that exercise may have the ability to regulate nitric oxide levels in skeletal muscle. The strengths of this study include the fact that it was the first study to compare differences in skeletal muscle nitric oxide levels post-doxorubicin administration, with and without acute exercise following dox administration. In terms of future directions, a control group should be implemented for comparison of nitric oxide levels in healthy tissues. Additionally, a longer experimental protocol with multiple exercise bouts and differences in exercise duration and intensity should be completed. More research in this realm has the potential to lead to better patient outcomes and improve the quality of life of those undergoing chemotherapy treatment. That concludes my presentation. Thank you for listening.